Good afternoon. We'll continue with our previous class. In the last class, we have stopped at DNA damage and in repair part. So we have already seen that cells must undergo continuous repair. Because of that, the damaged DNA will be causing continuous call for the repair of the DNA. So that is always there in the system. And the bases may be altered or lost or may form double-stranded beads. Chemicals and ultraviolet radiations damage the DNA in our body. If the damage is not repaired permanently, it can lead to cancers or it can lead to mutations that will lead to cancers. What are the various types of damages that a DNA is susceptible to? It can be during the replication errors. We have seen that replication errors are very common. There are many replication errors that would be happening in the proofreading, and all those things have to be done. So replication errors is one of the commonest cause for misalignment of the DNA. Then X-rays, UV light, alkylating agents, spontaneous reactions. In the free radical formation we have seen there is development of spontaneous reactions that will damage the cell wall. So in that also they will be damaging the DNA. Next, reactive oxygen species. So these are the things that will be damaging the DNA. Now what will the DNA do? Either it will do repair mechanism or it will surrender to the damage. It can surrender to these agents and get apoptosed, or mutations can be formed, or cancers, anything can result. And we have also covered that there are 4 types of repair mechanisms: mismatch repair, base excision repair, and nucleotide excision repair, and double strand break repair. We will individually see what all the various repair mechanisms. First, mismatch repair. The name itself is indicating there is a mismatch. The base pair rule has to be followed. That is, adenine has to bond with the thymine and the guanine should be forming with the cytosine. So it should be AT and GC. If this type of pairing does not occur, if a purine combines with a purine or pyrimidine combines with a pyrimidine, then you call it as a mismatch. A should go with T only. A should not bind with G or with C. So that is what is a mismatch. If the base pairing rule is not followed, that means there is a mismatch. And this type of mismatching is very common in the replication process. So when replication takes place, there are sometimes the nucleotides can undergo mismatches. So it corrects the error when DNA is copied. So this repair mechanism corrects the errors that occur during the replication process. Specific proteins scan newly synthesized DNA with reference to the methylated GATC sequences. This difference allows the enzyme to recognize the new strand. GATC nucleotides cut the mismatch. Then they are filled by ligase enzymes. We have two DNA. We have a DNA double helix. So this is one DNA and this is another DNA. Now, how will you know that which is daughter and which is a parent strand? Usually, the parent strand will be having methylation at GATC base pairs. Intermittently, on the parent strand, there are GATC base pairs will be there, and these are methylated. The parent strand is methylated, and this is recognized by special proteins called as mute proteins. The mute proteins recognize the mismatch and identify the methylated parent strands. The segment of the daughter strand is released. So this part is released. Now the polymerase fills the gap and ligase joins the newly synthesized. The polymerase fills the gap. The ligase will join the new edges and it will form the normal. So the mismatch that is present is repaired. So the DNA polymerase and DNA ligase, the mismatch that is present is repaired. So this is how the mismatch repair is done. This type of repair mechanism is seen in rectifying replication errors. Next, What will happen if this repair mechanism is defective? Usually the proteins that recognize the mismatch are MUT-S, MUT-C and MUT-H proteins. They usually recognize the mismatch. There is a disorder called as hereditary non-polyposis colon cancer, that is HNPCC, hereditary non-polyposis colon cancer. And this results when there is mutation of genes. They are named as HMSH2, HMSH1. So MSH1 and MSH2 genes, this is a gene that will be coding for this protein, MSH1 protein. So these two code for this MSH1 and 2 genes will be coding for the proteins that help in repair mechanism. Now whenever there is mutations of these two genes, the protein that repairs is not synthesized. As a result, it will lead to a cancer called as hereditary non-polyposis colon cancer. So what will happen in the colon? There will be development of polyps. There will be development of polyps because MSH1 and MSH2 genes are not producing the protein that is required to rectify the mismatch repair. Next we'll go into another one called as base excision repair. Depurination means loss of the property of a purine is called depurination. Depurination of DNA is a spontaneous process which occurs at a rate of 10,000 per cell per day. What do you mean by depurination? Depurination means the cytosine, adenine and guanine bases are spontaneously converted to form uracil, hypoxanthine and xanthine respectively. So cytosine will get converted to uracil, adenine will get converted to hypoxanthine, guanine will get converted to xanthine. So this phenomenon is called depurination. Like this. So this actually G is pairing with C, so it is not a mismatch, everything is fine here. But because of some because of some disturbance or anything, there is spontaneous deamination of cytosine. As a result of that, the cytosine is getting converted to uridine. It is getting converted to uridine. Now, so these are not normal constituents of the DNA. Actually, this should not be present in the DNA. That is not a normal constituent. So what will it do? A specific enzyme called as N-glycosylase can remove these abnormal bases. So these abnormal bases can be removed as such by means of this glycosylase enzyme. What will it do? It will remove the abnormal base, but the ribose and the phosphate that are attached to this nucleotide will be attached like that only. Next, now it will have a ribose. Actually, what we have, a nucleotide will be having a nitrogen base. It will have a ribose and a phosphate. These are compulsorily present. Now, by means of glycosylase, what has happened? Here, uridine was there. From the cytosine, it has got converted to uridine. Now, this uridine is removed, but still there is a sugar with no base, that is ribose is still present. So that is removed by deoxyribose phosphate ligase. So this deoxyphospholipase or endonuclease excise, any of the enzymes, either by the endonucleases or by deoxyribose phospholipase, that sugar with the phosphate moiety are removed. Now we are having a absolute blank place that doesn't, you can see the gap here, that short amount of the orange line that you can see. This is the sugar and ribose backbone. Now in this picture, that is missing, means that is gone. Now in the presence of polymerase and first polymerase, it will get the original nucleotide that has to be here, that is cytosine will be there. Then ligase will be joining both of the nucleotides so that the chain will be continuous. So spontaneous depurinization are rectified by means of base excision repair. Next is nucleotide excision repair. When do we see this nucleotide excision repair? It is an important mechanism by which the cell can prevent unwanted mutations by removing the vast majority of UV induced DNA damage. The UV induced DNA damage is rectified by nucleotide excision repair. There are 9 proteins that are involved in this nucleotide excision repair. Wrong bases are removed by endonucleases up to 24 to 32 bases around the long base. So here exactly the wrong base that is present is not removed. 24 to 32 base pairs around the wrong base are removed. So it is a sequence of 24 to 32 bases, nitrogen bases are being removed. This is how. Now it has identified these as the nucleotides that are problematic, that have got modified or anything. So they are not the normal ones. So what will it do? It will take up almost this stretch that constitutes up to 32 to 40, like a huge number of nucleotide. This much sequence is removed by endonuclease. So this endonuclease will remove that, and then once again the polymerase and ligase will synthesize the new one, and it is exactly complementary to the parent strand. So this is how the nucleotide excision repair occurs. Now, what is a clinical disorder that is associated if this nucleotide excision repair is not working properly? The clinical disorder called xeroderma pigmentosa results because of abnormality or inefficient repair. It is an autosomal recessive disorder. There is marked sensitivity to sunlight with formation of skin cancers and premature death. So the persons will have marked sensitivity to sunlight. This is mainly because of mutations in XP gene. So because this is expanded as xeroderma pigmentosa gene, and that is named from A2 gene. You have xeroderma pigmentosa A, B, C, D, E, F, G. So in this way you have proteins. Now mutation of any one of the protein is enough to prevent the repair. These proteins are involved in recognition and excision. So now mutation of any of these proteins occurs, the wrong nucleotide cannot be recognized and it cannot be excised. So this is how a person with xeroderma pigmentosa will appear. You can see there is marked sensitivity to the sun, and you can see the scales have formed. There are so many blisters and all that. This is a xeroderma pigmentosa. Next, double strand break repairs. Now double strand break repairs means in all the previous repair mechanisms we have seen one strand is normal, there is problem with the second strand, there is problem with the other strand. So it is taking the strand that is fine as a template and it is synthesizing the other strand, or you can say it is replicating the other strand. So previous 3 repair mechanisms that we have seen have had an intact, one strand was intact, the other strand was not intact. But in case of double strand break repair, what will happen in double strand break repair? There will be breakage of both the strands. Both the strands will be breaking. And these are very much dangerous. These result in genetic recombination which may lead to chromosomal translocation. They may result in genetic recombination which may lead to chromosomal translocation, broken chromosomes and finally cell death. Physiological processes of immunoglobulin rearrangement occurs by this mechanism. Physiological processes of this will be occurring by this mechanism. And so that is physiological double strand repair mechanism. But the influence of ionizing radiation, cancer drugs and free radicals, they can also bring about double strand break repair. Now we'll just look into what all repair mechanisms which we have come across till now. Mismatch repair, copying errors, single base pair or 2 to 5 base unpaired loops. So methylated directed strands are cut, exonuclease digestion and replacement. Next, base excision repair. The commonest cause is chemicals, radiations and all that. And this is done by N-glycosidase. It will remove that base and then that A basic sugar is removed and new nucleotide is added. Nucleotide excision repair. This occurs because of chemical or radiation damage. This involves in removal of 30 to 40 nucleotides that are adjacent to this mismatched nucleotide. So 30 to 40. So a large stretch of nucleotides are excised. Next, double strand repairs. These are physiological in case of immunoglobulin synthesis, or these can be because of ionizing radiation, chemotherapy and free radicals. And how is it rectified? By means of synapses, by means of unwinding, by alignment or by ligation. So both of them are fused, some part is unwind and it is aligned and then it is ligated. This is how the double strand break repair is rectified. Double strand break repair is done. Next, we have already seen, so we have covered the replication process. Then we have seen if there is any problem with the replication, how the body repairs or rectifies those defects. Now the genome is prepared. Now the genome has to be organized properly to a DNA. It has to be organized, and this organization we have already seen that histones will be helping in formation of that. So this octamer of histones, the DNA will bind around the octamer of histones, and in between these two there is H1 is present that will prevent the collapse of these nucleosomes. So you call it as a nucleosomes. The DNA combined with the DNA that is wound round around the histones, this whole complex you call it as a nucleosome. So it will prevent the collapse of the nucleosomes. Now, so this is a naked DNA. It is organized around the histones to form nucleosomes. So these are called as nucleosomes. Many nucleosomes will combine to form the filaments. These filaments are called as nucleofilaments, and these nucleofilaments further coil to form chromatin or chromosomes. So this is how the DNA is organized. Now we'll see one more new thing that is called as recombination of DNA. This is not recombinant DNA. Recombinant DNA is the one that is synthesized. It is a synthetic DNA. This is the mechanism by which DNA undergoes recombination by itself or because of some stimulus. So that we call it as recombinant DNA. Now, what do you mean by recombination? Recombination, as the name indicates, recombination means rearrangement of genetic material in and among DNA molecules. Rearrangement of genetic material in and among the DNA molecules encompass a variety of processes which is called as genetic recombination. So rearrangement of genetic material. What are the types of recombinations? The various types of recombinations are homologous recombination, of which the most common is Holiday model, site-specific recombination, and DNA transposition. These are the three types of recombinations that we know. What is homologous recombination? Recombination of two different DNA molecules or two segments of same molecule which share a homologous sequence. So this is very important because they have to share a homologous sequence. Then only you can say it as a recombinant. Then only you call it as a homologous recombination. It occurs during meiosis time. In the meiosis time it occurs. So this is the one that helps in maintaining the genetic diversity. So genetic diversity, because the siblings are different from one another. Though the parents, the genome that they attain from the parents is same, but the variations in the siblings is because of the genetic diversity. So during the meiosis time, what will happen? There will be mix up of recombination. So some part of it will randomly join with other part of the chromosome. That is the reason for the genetic diversity. Now, of the holiday, as I told you, you have we have 2 to 3 types of the homologous recombination, but the commonest one is holiday model. So we'll be knowing details about this holiday model only. The two homologous chromosomes come closer and they get aligned. This results in two aligned DNA duplexes in this way. So two DNA that have to be aligned. So one is in blue color, the other one is in pink color. So this is a nucleotide. So this is a DNA duplex. So this is a duplex, like alpha helix. It has got, this is one strand, this is one strand. So don't think that this is a single strand. It is a double strand. These two blue lines are the strands. This is going from 5' to 3'. The lower blue line is going from this direction. Now the two pink lines are two strands of another DNA. Now we can label it as capital A and capital B, and this is as small a and small b. Now if the recombination has to take place between these two DNA duplexes, what will happen? Both of them, they get rid of their coils, or they get rid of the helix structure, and they align straight. In that way they will be aligning straight. Now, the strands of each duplex partly unwind and invade into the opposite direction to form a two-strand cross between the DNA molecules. So, this is one helix and this is another helix. Now, these two helix have opened up and they have become straight. They have unwound and they have become straight. Now, what will happen? One part will invade into the another part. The endonuclease brings about a small nick in one of the strands. This is a double helical strand, this is a double helical strand. So, from the double strand, one strand will break, and here also one strand will break. Now, this occurs in such a way that after this strand will combine with, now there is a nick here, there is a nick here. This strand will combine with the strand that is present below. This strand will align with the strand that is present above. So, there is crossing between the two DNA molecules. There will be a cross between the two DNA molecules. Now, once they cross, the gap between both of them is lysed. It is joined by means of ligase. Ligands will join both of them. This phenomena is referred to as branch migration, and this results in the formation of a DNA that is called as heteroduplex DNA. So this we have seen. Now the blue DNA, that is capital AB DNA, has merged with this DNA. Now this DNA is merged with this DNA. So this is called as branch migration, and from this cross, when they align properly, the structure that is formed is called as heteroduplex. So this structure is called as heteroduplex. Now this is how, when the heteroduplex rotates, this is how the heteroduplex appears. This is how it is shown. So what will happen? There is a crossing over, and the crossing over is called as branch migration. The product that we get from the branch migration, you call it as heteroduplex. Now, 4 strands of DNA are joined by a single crossover point, and it can rotate to create a 4 strand. So that is why it is called as holiday junction. Now here you can see that 4 strands are joined in this way to a center point. 4 strands are joined together, and you call it as a holiday junction. The enzyme nuclease forms a nick at the DNA duplexes. So this is a holiday joint. Now this endonuclease will cause a nick between them in this way, and the nick will separate the 2 recombinant strands. The enzyme endonuclease forms a nick at the DNA duplexes. The parent strands are cut vertically. The flanking genes get exchanged due to the recombination. Now what you can see here, we can see that there is exchange of genome, complete exchange of genome. So initially we had only blue and we had only pink. Now there is combination of both blue and pink. There is combination of both blue and pink. So this is called as homologous type of recombination, and the commonest phenomenon of homologous recombination is called as holiday junction or holiday model. Next is site-specific recombination. The site-specific recombination, it occurs when there is exchange only at a defined sequence. When the exchange is exactly at a defined sequence, then there is site-specific recombination. The enzyme that is required in this process is called as recombinase. Immunoglobulin gene segmentation usually occurs by site-specific recombination. Now this is a gene that codes for heavy chain of the immunoglobulins. Immunoglobulins will be having 2 chains. You have a light chain and a heavy chain. This is the gene that is coding for the heavy chain. Now in between, some intervening genes are the ones that are not required. So what will happen? They are removed. So it's site specific. So to these specific site they are removed. Now there is recombination of the D and this J. So in between D and J interventions, and there are many 4 Ds and 4 Js. So 2 Ds and 3 Js are gone, and D and J are uniting. So there is recombination of DJ exons forming a DJ combination. Now removal of unwanted V, and there is no requirement for so many V here. So the unwanted V has to be removed. So these unwanted V's are removed, and removal of V and DJ exons, VDJ complex is formed. So that is called as VDJ recombination. So the gene was of this length which had many unwanted genes, and that are being removed and recombination has taken place. So this is a physiological phenomena in which there is formation of there is formation of immunoglobulins, that is heavy chain of immunoglobulins are formed by this mechanism. Next, DNA transposition. The transposons or the small segment of DNA with remarkable capacity to move to same or another chromosomes. They are called as transposons. They have capacity to move around, that is they can get incorporated into any of the DNA, either of the same DNA or of another chromosome. Retrotransposition. Retrotransposition is a phenomenon in which The DNA is formed by reverse transcriptase. So the DNA is formed from reverse transcriptase, that is from mRNA of the transposon. So this is a target DNA and this is a target site. So this is a target site. What will happen? This is a transposon that has come. Now this transposon will get combined with the target site. Now it will identify to what site it has to go. So it will identify this site and it has to attack this site. So what will happen? It will come in the presence of endonucleotide. A small gap is created and this transposon will get interposed, or it will get transposed into the target DNA. So now it has freely entered into the target DNA and the transposon has united with the target DNA, and any gaps are present that are joined by means of ligases. So this is how the transposon will be getting incorporated into the target DNA. Now, what is the significance of recombination? We have seen there is a homologous type of recombination where the same set will occur, and that is the commonest thing for genetic variation. Next, we have seen site specific, that is common for immunoglobulins. Next, we have seen the transposition where another small DNA independent transposon that is not part of any of the DNA, that segment will come and it will get incorporated into the DNA. So there are pros and cons of having the recombination. Most important, why recombination occurs in the body is, we can explain it in other way around, why there is genetic diversity. The genetic diversity is mainly because of the DNA recombination that happens normally. And this recombination is also very important. I told you in case of double strand repairs, in the previous 3 repair mechanisms, there was a template because of the 2 strands, only 1 strand was damaged. So there is another strand that can be used, the parent strand, the 1 strand that was not damaged, that can be used as a template to and so that the daughter strand or the another strand can be repaired. But in case of double strand breaks, that is much more dangerous and much more crucial. So in case of double strand breaks, what will happen? There is no parent one or no intact strain that will get. So in those cases, what will happen? This recombination will help in the repair because body cannot leave the broken double strands like that. So they are combined by this type of recombination. So it is one of the type of recombination mechanism. Next, it helps in regulating the gene expression. So how it regulates the gene expression, detailedly we'll see when we are talking about the regulation of gene expression. But that is also very important. I can give you the example of this immunoglobulins that we have seen. So immunoglobulins, we have seen that in site-specific recombination where that intervening genes are removed. So that is how that gene is regulated. That is one of the example of gene expression. Next, programmed gene arrangement. The one that we have seen for the immunoglobulins. The genes are arranged subsequently or in a sequence manner. So that is called as programmed gene rearrangement. So this is about the recombination. We are done with the replication process of both prokaryotes and eukaryotes. Then we have seen the DNA repair mechanisms, and we have seen a little bit about the recombination. This is recombination that occurs. Recombinant DNA is the one that is synthesized outside the body. This is a recombination that takes place. Now I can see only 22 people are there in the live chat. And I want to give you an assignment. Now we are not having any we don't know about the internal assessments, how we are going to take up assessments and all that. So to start with, I want to give you an assignment in which I want you people to write a tabular column differentiating eukaryotic and prokaryotic replication. You have to draw a table to differentiate eukaryotic and prokaryotic replication. From the first, like from origin of replication till the termination of replication, how prokaryotes and eukaryotes vary. And that I'll put the question that has to be like, I'll frame the question properly and I'll put it up in WhatsApp of your CR. And I'll tell you like, and you have to write it. You don't do it on Excel or some, you don't do it on MS Word like that. You have to write it on a paper. You have to send it to the mail. So what numbers to what email ID you have to send, I'll post it to your WhatsApp of your CR. You follow from that. And this has to be done by the Saturday. Now today is Tuesday. By Saturday I want it to be done. By today evening I'll post the question and everything properly. So this is the end of DNA replication.